Hello, welcome to our presentation on the state of research specifically targeted to understanding SPG3A, a form of hereditary spastic paraplegia, and possible approaches to therapy. Our hope is that this is going to be the first of several updates over the next couple of years on this promising area of research. I'm Joe Avellone. I'm the president of the Carter Foundation for Neurologic Research, but more importantly, I'm the grandfather of Carter, a wonderfully happy 7-year-old who lives every day with this condition. He is a wonder to behold, and as I know all of your children are as well. I'd also like to introduce Dr. Craig Blackstone. He's a physician and research scientist who is a world leader in focusing on hereditary spastic paraplegias and SPG3A and SPG4 in particular. He's a director in the motor disorder division of the Division of Neurology at Massachusetts General Hospital and a professor at Harvard Medical School. But prior to this, he had spent almost 20 years at NIH studying just these kinds of disorders. So we're very lucky to have him as our senior advisor at the Carter Foundation. Welcome, Dr. Blackstone.
Thank you, Joe. It's my pleasure to be here.
Finally, I'd like to thank our hosts, AJ and Juan, who have a wonderful, happy little girl, Emily, also living with SPG3A, and who have founded and run the website, Kids with SPG3A. Next slide, please. And the next. As I mentioned, the Carter Foundation is focused on finding therapies for children with HSP, particularly SPG3A and 4. We're trying to close the gap between the current basic science understanding of these diseases, which is considerable, and the ability to develop human therapies. Filling the gap means taking off from the basic science that we know and testing therapeutic theories in animal models. Once we have shown a therapeutic response that is measurable in an animal model, it's very likely we would then be able to move on to development of drugs and other therapies, enlisting biopharmaceutical companies with an interest in these kinds of diseases. So bridging this gap is called translational medicine, and that is our current focus. Next slide. Currently, based on much of the work done by Dr. Blackstone and colleagues at NIH and some other researchers around the world, we are focusing in on 4 possible approaches to therapy. As you can see listed there, lipid modulation, BMP signaling inhibition, ASO therapy, and advanced gene editing. Next slide. So let's start with lipid modulation. Dr. Blackstone, could you give us an idea of the scientific rationale for this one and what studies we will be performing in the next year or so to study it?
Yeah, sure, Joe. It'd be my pleasure. One of the things about this hereditary spastic paraplegias as a group is that there's many different genetic forms. And over the last couple of decades, we've identified almost 100 different genes that can cause different forms of hereditary spastic paraplegia. And when we look at those genes and the proteins they encode, we see certain themes appear. And one of those is around an organelle in the cells called an endoplasmic reticulum. And that organelle within cells has lots of distinct functions. But one of the key ones is the formation of lipids and their distribution throughout the cell. And remember, a neuron is a very, very large cell, far larger than most other cells in the body. And the disease, this hereditary spastic paraplegia, specifically targets these really, really long neurons. So that's why it's sort of unique to the nervous system, a lot of the symptoms that patients have. Now, several other genes that encode different forms of hereditary spastic paraplegia are actually lipid metabolizing enzymes themselves, some of them localized to the endoplasmic reticulum, some in other parts of the cell. So when we look at the hereditary spastic paraplegias as a whole, there's well over a dozen different forms that seem to converge on this theme. And in particular, of course, SPG3A and SPG4, which again, to explain what those mean, those are just when they initially identified the HSPs, they just numbered them in order of discovery. So 3A and 4 are just 2 forms, but they happen to interact with each other in this pathway. So we think this is a particularly promising pathway. Now, when we look at the specific things like a liver X receptor agonist, that's a very specific type of pathway that regulates lipids within the cell. And that's, of course, based on, as exactly as you said, Joe, several groups have been studying different aspects of this pathway and have identified that as a potential therapeutic target. And as you also said, one of the things that we want to do as we do translational research and bring this to patients is we want to identify specific targets within the cell that we can create molecules against, drugs essentially, that could ultimately help treat patients with these disorders. Now, so in your next stage, you emphasize that we want to look at these agonists in more detail in different cellular systems. Some of those can be mouse models, they can be fly models, or they can be human neuron models. And I'll spend a minute just describing what that is. You can actually take skin cells from patients with disorders and through a series of manipulations, actually create stem cells that can then be differentiated into nerve cells. So you have the ability to look at a nerve cell from a patient, essentially, and test it. And that gives you the idea is if there's an abnormality in that cell, can we correct it with these drugs? So one of the key projects for the next year is using these different agonists to see if we can correct defects in these human nerve cell models, as well as in other models such as mice and flies and other types of what we call in vivo models that are also important aspects of ensuring that drugs work not only in cell culture systems, but also in organisms themselves. Much like, and ultimately, of course, the goal is to determine whether it's safe and effective for people.
Excellent. Thank you, Dr. Blackstone. So go to the next slide, please. The next area of study is called BMP signaling inhibition. So that scientific title is not very self-explanatory. Dr. Blackstone, could we ask you about this rationale and what stage of study that we're going to be endeavoring to do this next year or so?
Of course. So bone morphogenetic protein signaling, as you said, it's not so obvious, and it's actually a part of a larger signaling sort of pathway known as the transforming growth factor beta signaling pathway. And those are commonly studied for lots of different disorders, including development, abnormal development, or even in things like cancers. So it's a pathway that's been studied for a very long time. But years ago, many years ago, actually, when people were studying some of these hereditary spastic paraplegia genes, they found that several of them were actually involved in this pathway. And that was a little surprising because it wasn't so obvious. And one thing to emphasize is just because the word bone is in it, it doesn't mean it only functions in bone morphogenesis or production. Yeah. That was just the first place it was found, so they gave it that name. But since then, it's been found to be involved in a lot of other pathways within certain types of cells. And there's actually been strong evidence over the years that many of the HSP proteins actually function as inhibitors of this pathway. So in other words, if you have the normal function SPG3A protein, which is known as elastin, it would actually inhibit this pathway. And our thought is that some of these mutations might actually essentially remove that inhibition so that the pathway is too active. And you might think, well, isn't more better? And it's not, because a lot of as we develop, a lot of our pathways get turned on and off throughout development. And sometimes having something continuing to be active is not a good thing. And fortunately, because these have been studied for so long, we know exactly how to see if our inhibitors are effective. So in a sense, for both SPG3A and SPG4, which have both been implicated in this pathway, you can imagine that we would say, okay, the protein isn't functioning properly. Let's give an inhibitor that does the same thing. And therefore, we can correct this pathway, and maybe that will improve symptoms. So that's sort of the rationale for doing this. And again, this is a pathway that's been studied for decades. But one thing we did want to emphasize is that for children with the SPG3A and SPG4 mutations that we're describing, their symptoms tend to be earlier onset and more severe. So even though we know that some of these pathways are important in sort of the traditional forms of these disorders, we want to do two things. First, we want to confirm that they're also affected to the same degree and in the same way in these earlier onset, more severe forms. And then second, we want to test this in different systems. We want to move this idea forward to see if it's something viable for the treatment of, again, probably not only these children, but also a larger number of people with different forms of hereditary spastic paraplegia. And much like the previous slide, or the previous studies of liver X receptor agonists or this lipid pathway studies, is we would do several of these investigations, both in human models, which are these induced pluripotent stem cell or iPSC models, and also in what we call in vivo models, flies that have modeled the same type of mutation, or mouse models that have been created. And again, in all cases, these have been selected to model the specific mutation or mutations that give rise to the symptoms of these disorders.
Thank you, Dr. Blackstone. So if we go to the next slide, the next therapeutic approach that we're going to study is called ASO therapy. It's actually called ANTISENSE oligonucleotide therapy, which is a rather complicated name as well. This one uses special kinds of RNA to work with the actual production of proteins in the neurons, specifically the elastin-1 protein that is involved with SPG3A. So Dr. Blackstone, can you tell us what this is all about and the kinds of studies that we are pursuing to explore it?
Sure. And this one requires a little bit of understanding of genetics of these disorders. So one of the features of the children who have these particularly early onset and severe forms of SPG and SPG3A and SPG4 are that these are not mutations that they inherited from their parents. These are mutations that just occurred spontaneously. And therefore, they just appeared in one of the two genes. So for most of our genes in our body, we have two copies, one from each parent. But you can also you don't have to just inherit them. There can just be mutations that occur spontaneously during development or before that. And then you have essentially a mutation that has occurred that wasn't in from either of your parents. And sometimes these can cause disease. Many times they just make us different. But occasionally, in very rare cases, they cause disease. And that's what's happened in these children. So if you think about it, we have what we'll call a bad gene because it's mutated. And then we have a good gene that's completely normal. And because these disorders are what we call dominant disorders, in all the patients with SPG3A and SPG4, they have a normal gene. So one thought is, well, what if we just get rid of the bad copy? People still have a normal copy. And even if they have a little bit of symptoms from that, maybe they would be a lot more mild, a lot less severe, in other words, than having this abnormal gene. So it emphasizes the point that sometimes it's better not to have a mutant protein versus, having it. Sometimes its presence isn't just that it doesn't have the right function, but that its presence is toxic. And that's what, as you have on your slide here, it's a toxic gain. It's gained some new abnormal function. And if we remove that, we still have a normal gene that might be able to, fill in most of the needs. And that might normalize the functions of the cells and ultimately reduce the symptoms. And ASO therapies have been in the news a lot because they've been found to be very effective for certain disorders. So we know a lot about how to do this and how to deliver them. And we're getting more and more experience with this in the nervous system. And the nervous system, again, can be a very challenging area to treat because it's not easy to get to with either drugs or some of these therapies. But since we already know that it's been effective and that it's been safe in a lot of other disorders, we know that this is a promising approach. And from a strategic point, the advantage of this is, as we've discussed already, we have a couple ideas of pathways that we can pursue. For this one, we don't really have to know exactly what function is involved, or it's sort of like we can get away with just knowing that we have a bad protein and we want to remove it. And by removing it, then hopefully we've restored a lot of different cellular functions. We don't have to target one specific pathway. Hopefully this would have the effect of potentially affecting the two pathways we discussed, as well as other ones that we may not even know about. So it's a different way to approach things. But I think from a strategic point of view, it's very good to approach these disorders with several different types of therapeutic options. And of course, this is also done much like the same studies. These can be modeled in cells from patients. They can also be modeled in different animal organisms. And one thing I would emphasize about this, and I think we'll talk about it with some of the later ideas, is that to do this, one of the models that we created in the in the mice was actually using a portion of the human DNA sequence, so that the targets that we're using are relevant for the human sequence, not just the mouse sequence, because those sequences can vary from species to species in small ways, but those small differences might be important in terms of some of these, what we would call, RNA-based approaches, where we're actually targeting, sort of the transcript that actually produces the protein. So I think, again, this is a very good way to do it, because from a strategic point of view, we're targeting a very different aspect of the disease than in the two previous areas that we discussed.
Wonderful. Thank you very much. Next slide, please. The final area that we're exploring is gene editing. This is a very specific kind of gene editing that is starting to be used for these very small gene mutations, which are called missense mutations. So these advanced techniques can effectively splice the DNA in the very exact place where this missense has occurred and insert the normal sequence. So of course, this is more advanced and is going to take a bit longer to explore, but absolutely worth doing. So we are getting started on that with the same kind of mouse model that Dr. Blackstone just described. Can you explain this in a little bit more detail in the kinds of experiments that we are considering this year?
Sure. So again, a lot of people may be familiar with this, or they may not know that they're familiar with it, but they've probably seen it in the news, is that there's a technology called CRISPR, which essentially is using a set of tools to edit genes. So if you imagine you have an abnormality in a gene, you might think, well, the best way to do this is, why don't we just go in and fix that abnormality, and then we're done. The gene's back to normal. It can do its normal function, and that's essentially what these methodologies are able to accomplish. But there's a lot of challenges to it technically, as well as just in terms of delivery. You have to actually just go and fix this one nucleotide. And remember, we have billions of nucleotides. How do we target this one change very specifically? And second is, we have to get it to the right cells, and that's not so easy. In a culture dish, it might be, but if you think about a human, where you have your brain has developed over all these years, and now you're trying to go and fix it in all these cells, you have to be able to get it there, and that's not so easy. So a lot of the technologies that have been developed, and again, as Joe has listed here, with Dr. David Liu's lab at the Broad Institute and Harvard University in Cambridge, Massachusetts, they've developed a lot of gene editing tools that might be applicable to the treatment of disease. And some of these are based on CRISPR methodologies, which again have been widely studied, and some he's pioneered himself that are based on chemical biology approaches. And the hope is that we can use these tools to very specifically fix the specific one nucleotide gene mutation that underlies these disorders. Now, the challenge, of course, is you have to have a different one of these tools for each type of mutation. Whereas with ASOs, we can find other ways to deplete the mutant gene. We can target other parts of the molecule. Here, we have to actually go right for the specific changes. So it would be truly sort of precision or personalized medicine, and that has its own challenges. We'd have to make many of these and test them. But for most of the children with these disorders, they have there's in both cases there's one mutation that seems to be more common than others. So it's certainly a reasonable way to approach it and see if this is working. So there's two big challenges. First is, can we precisely fix this genetic defect, and can we find a way to deliver the tools that do this to enough of the cells that we can fix it? Now, one of the great advantages of this is, in theory, once we do this, if it's fixed, we don't have to do it again. Whereas with a lot of the other both medical therapies as well as ASOs, they have to be given repeatedly over time. So this, though it's the most challenging technically, and is really sort of the most cutting edge technologically, it has the advantage that it has the potential of being very definitive, in that once we fix it, we could say, okay, to some degree, we're done. We fixed the problem. And we don't even really have to understand the mechanism. We just know that we've gone to the very sort of root cause of the disorder and fixed that. And then, in theory, all of the things that occur downstream of that process should be resolved. So though it's the riskiest in many ways, just because of its difficulty, it has the ability to, or the potential, to be a definitive therapy.
Thank you, Dr. Blackstone. That was very helpful. If we go to the next slide, please. Just to summarize, the Carter Foundation is trying to close the translational medicine gap between our basic science understanding of these conditions and the ability to develop therapies ultimately to be used in our children. And in 2023, we're focused on 4 potential therapeutic approaches that we discussed today. The first was lipid modulation. The second was BMP signaling pathway inhibition. The third was so-called ASO therapy. And then finally, advanced gene editing therapy. The good news is that we're in the translational medicine phase of our research, working in animal models. The caution is that science can often go in many different directions, and certainly Craig, through his whole career, knows that more than any of us. However, the more we study, the more we learn. So thank you very much for your time today. We look forward to providing more updates as we get farther along in our research over the next couple of years. And I also want to thank Dr. Craig Blackstone for all his pioneering research to get us to this point and for being the advisor over this set of experiments as we go forward. And finally, in closing, I'd like to thank our hosts, AJ and Juan, and thank them for all the work they've done in bringing this HSP SPG3A community together.